During interphase, the cell organelles double in number, DNA replicates, and protein synthesis occurs. Mitosis is the division of the nucleus. The genetic material is duplicated so that only one copy is passed on to the daughter cells. In cytokinesis, a cleavage furrow forms, splitting the cell in two and pinching off the separated nuclei. Mitosis is the process by which cells multiply. It enables an organism's cells to reproduce and regenerate tissue in the body. In G1, the first substage of interphase, the cell starts to grow. Then in the synthesis phase, the cell continues growing as it copies its chromosomes. In G2, the cell grows more as it completes the necessary preparations for cell division. Prophase, the nucleoli disappear and the mitotic spindle begins to form. Asters form extending from the microtubules. The centrosomes start to move away from each other, propelled by the lengthening microtubules between them. Metaphase, the nuclear envelope fragments. Each of the two chromatids now has a kinetochore, which is a protein structure at the centromere. Some of the microtubules attach to the kinetochores, jerking the chromosomes back and forth. In metaphase, the chromosomes lie on the metaphase plate, held there by centromeres. For each of the chromosomes, the kinetochores of the sister chromatids are attached to the kinetochore microtubules coming from the opposite poles. In anaphase, the sister chromatids part suddenly. These new chromosomes move towards opposite ends of the cell as the kinetochore microtubules shorten. The cell elongates as the non-kinetochore microtubules lengthen. In telophase, two daughter nuclei form, the nuclear envelope arises, and the chromosomes condense, completing mitosis. Cytokinesis divides the cytoplasm by forming a cleavage furrow, pinching the cell into two daughter cells. Mitosis results in two diploid daughter cells that are genetically identical to each other and their parent cell. The DNA is duplicated and the chromosome number is doubled. Meiosis reduces the number of chromosome sets from diploid to haploid within its two divisions. Because crossing over occurs, it brings together a new gene combination of chromosomes for future generation. Meiosis 1 is the first step in the division of our sex cells. In prophase, crossing over occurs. The chromosomes in each homologue separate slowly, then the microtubules from each pole attach to the two kinetochores, moving the homologous pairs towards the metaphase plate. In metaphase 1, the homologous pairs are now arranged at the metaphase plate. Both chromatids of one homologue are attached to the kinetochore microtubules from opposite poles. Anaphase 1 is responsible for the breakdown of proteins gluing the chromatids together. This allows the homologues to separate and move towards opposite poles of the cell. In telophase 1, each half of the cell has a complete haploid set of chromosomes, composed of two sister chromatids. Cytokinesis occurs simultaneously with telophase 1, forming two haploid daughter cells. In prophase 2, a spindle apparatus forms and each of the chromosomes start moving towards the metaphase 2 plate. In metaphase 2, the chromosomes are positioned on the metaphase plate. Sister chromatids of each chromosome are not genetically identical, and the kinetochores of the sister chromatids are attached to the microtubules extending from the opposite poles. In anaphase 2, the proteins holding the sister chromatids together break off, allowing the chromatids to separate. The chromatids move towards opposite poles as individual chromosomes. Telophase 2, the nuclei form, chromosomes begin condensing, and cytokinesis occurs. At the end of meiosis 1 and 2, 4 daughter cells are produced, each genetically distinct from the other daughter cells and the parent cell. Meiosis 1 is the separation of homologous chromosomes. Crossing over occurs here and results in two haploid daughter cells, consisting of two sister chromatids. Meiosis 2 is the separation of sister chromatids. This occurs simultaneously to the two new daughter cells, resulting in two more haploid daughter cells. Meiosis divides 2 times within its cycle, resulting in 4 daughter cells, while mitosis divides once and ends with 2 daughter cells. In meiosis, crossing over occurs, whereas crossing over doesn't exist in mitosis. Daughter cells in mitosis are diploid, while daughter cells in meiosis are haploid. Both cycles include the division of a cell, while mitosis divides only somatic cells, and meiosis divides gametes.